Greetings and welcome. Today I'm going to be finishing up the bacteria and archaea. This is part 2 for the Natural Science 102 course. So here's just a list of things we're going to cover today. Cell wall differences between archaea and bacteria, reproduction and genetic transfer. We won't talk too much about the plague, but there's a video that I refer you to in the description below, and some information about viruses. So this is a picture of the plasma membrane, basically the cell wall, okay, of these bacteria and archaea. So the prokaryotic plasma membrane is a thin lipid bilayer, as you recall, that completely surrounds the cell, and it separates the inside from the outside. Its selectivity, permeable nature, keeps ions, proteins, and other molecules within the cell and prevents them from diffusing into the extracellular environment, which means the outside of the cell, while other molecules may move through the membrane, okay? So again, recall that the general structure of a cell membrane is a phospholipid bilayer composed of 2 layers of lipid molecules. The archaea cell membranes are phytanol chains linked to a glycerol replace the fatty acids linked to glycerol in bacterial membranes. And there's a little figure there. So in number 1, you can see how there's an ether linkage. That's in the archaea. And number 2 is pointing to the typical phospholipids of bacteria, okay, and your cells that make up your body. Now, in archaea cell membranes, the phytanol chains are linked to glycerols, replace the fatty acids linked to glycerol in bacterial membranes. So some archaeal membranes are actually lipid monolayers instead of bilayers, but the main difference here I just want to point out is this figure here. Archaeal phospholipids differ from those found in bacteria and eukarya in 2 ways, okay? First, they have branched phytanol side chains instead of linear ones. You can see how in the bottom there's just this zigzag that goes straight across, okay? And in the archaea, in the pink, oval there, you can see how there's that, it doesn't look like just a straight zigzag. There's those little lines coming off of it, okay? We call that a branched side chain. Number 2, an ether bond instead of an ester bond connects the lipid to the glycerol, okay? Down here where it says number 2, it says ether linkage, and it's pointing to the archaea membrane, so it says C-O in that little yellow circle. And then down below we have an extra oxygen there. That's called an ester, okay? The bottom line is that, and then there's some pictorial representations down on the bottom of what the phospholipid bilayer looks like for the archaea on the right. They're kind of joined in together. They're very strongly intercalated, it looks like, and it's a much stronger lipid bilayer. On the left, you have bacteria, which is a lipid bilayer that's just kind of loosely held together, okay? So the archaea lipid bilayer is more sealed, it's stronger, it's a very strong bond between the 2 halves of the lipid bilayer, and this makes them extremophiles, as you recall. Let's talk about reproduction. Now, reproduction in prokaryotes is asexual, okay, and that means that they themselves just divide and create copies of themselves, and it usually takes place by binary fission, okay? So here's a little figure showing the binary fission. So again, recall that DNA of a prokaryote is a single circular chromosome. So that's shown with a little blue circle here, okay? It's within the cell, all right? You've got the cell wall in darker brown, and then you have the cytoplasm, which is all the juice and electrolytes from the bacteria or archaea. And then you have the cytoplasmic membrane, which is really hard to see in this figure. Now, prokaryotes do not undergo mitosis. Instead, the chromosome is replicated and 2 resulting copies separate from one another due to the growth of the cell. The prokaryote, now enlarged, is pinched inward at its equator. The technical word for that is called the septum, but basically it just starts pinching off and then they come apart, okay? And then this results in two cells which are clones, which are separate from another. Clones mean, exact copies, okay? So binary fission does not provide an opportunity for genetic recombination or genetic diversity because the daughter cells are going to be completely identical to the parent cell. But prokaryotes can share genes by three other mechanisms. The first way is by transformation. So first of all, I'll back up. There are several gene transfer mechanisms that allow bacteria to change their genes. Here, number one, in transformation, the prokaryote takes in DNA shed by other prokaryotes into its environment. If a non-pathogenic bacteria takes up DNA for a toxic gene from a pathogen and incorporates the new DNA into its own chromosome, it too may become pathogenic, okay? So think of the green little circle here as the bad influence on the DNA there shown in purple. It takes that in there and it just takes over the cell, not completely, but it gives it a different personality. Think about it. Remember, DNA is the blueprint for what types of proteins and sugars and things are expressed by the bacteria. And so if the green part is a bad pathogenic gene, then it can make this bacteria pathogenic, okay? Harmful to whatever organism it might infect. Number two, transduction. So in transduction, bacteriophages, which are the viruses that infect bacteria, okay? So key word, bacteriophages are specific viruses that only infect DNA. All right? So in transduction, bacteriophages, it's a virus here shown in blue, I guess we could call that color up there on the top. It can move short pieces of chromosomal DNA from one bacteria to another. That green little line is some DNA, okay? Transduction results in a recombinant organism. Okay, so the organism that results there has that little green part in it. So it has DNA that was originally in it, the purple, and then it's combined with some green. So we call that a recombinant organism. Now, you might be interested to know this same technology is purposefully used to create transgenic crops by introducing external genes into soybeans to make them, for example, more resistant to pests or more productive and yielding more beans per acre, okay? Archaea also have viruses that may translocate genetic material from one individual to another, okay? So this is not just for bacteria. Number three, conjugation. So in conjugation, DNA is transferred from one prokaryote to another by means of a pilus, okay? Which brings the organisms into contact with one another and provides a channel for transfer of DNA. Pilus, remember, are those hair-like little features on the outside of bacteria that allow them to aggregate and come together and recognize each other and stick to each other or other surfaces, okay? Now, the DNA that's transferred can be in the form of a plasmid, which is a circular part of DNA, or as a composite molecule containing both plasmid and chromosomal DNA. Now, all these three processes of DNA exchange are responsible for introducing or changing the genes of bacteria and archaea. So basically, if a bacteria is always dividing and creating exact copies of itself, the question comes out, well, how does a staph, a staphylococcus, which is, become staph resistant to medications? Or we've heard about MRSA, different bacteria become resistant to antibiotics that can kill bacteria. And well, this is one of the mechanisms, okay? They develop genetic defects or mutations that allow them to be resistant. All right. I don't want to show you inline the video from YouTube, which is from the National Geographic, but this is about the plague. The plague is a bacterial infection. And there were 4 plagues in the past, and you can watch the video, click on the link in the video description below, or if you're taking my class, just click on that link through the Canvas course, okay? Now, where do viruses come from? What do they look like, and how were they discovered? Well, first, let's talk about the discovery of viruses. Viruses were first discovered after the development of a porcelain filter. It's called the Chamberlain-Pasteur filter, okay? And this can remove all bacteria that's visible in the microscope from any liquid sample. Now, in 1886, Adolf Meyer demonstrated that a disease of tobacco plants called the tobacco mosaic disease could be transferred from a diseased plant to a healthy one via liquid plant extracts. In 1892, Dmitry Ivanovsky showed that this disease could be transmitted in this way even after the Chamberlain-Pasteur filter had removed all visible bacteria or viable bacteria from the extract. Still, it was many years before it was proved that these filterable infectious agents were not simply very small bacteria, but were actually a new type of very small disease-causing particle. And we call these particles varions. V-I-R-I-O-N-S, virion, okay? Most virions, or single virus particles, are very small, about 20 to 250 nanometers in diameter, and these are too small to be seen under a microscope, okay? It was not until the development of the electron microscope in the late 1930s that scientists got their first good view of the structure of the tobacco mosaic virus, shown here in the figure. The surface structure of varions can be observed by both scanning and transmission electron microscopy, whereas the internal structure of the virus can only be observed in images from a transmission electron microscope. So a special kind of microscope that was high-tech. It wasn't invented until the 1930s, okay? Now, the use of electron microscopy and other technologies has allowed for the discovery of many viruses of all types of living organisms. So Dutch elm disease, for example, is caused by a virus. COVID, we call it, is caused by the SARS-CoV-2 virus, and so on, okay? And the common cold. Now, where did viruses come from? This is still under debate and a very active study. This figure, I'm not going to go into it, it's probably too small to even see on your computer or phone, is a snapshot of a figure from a 2019 publication in Nature. It is a very active area of research, okay? Although biologists have a significant amount of knowledge about how present-day viruses mutate and adapt, much less is known about how viruses originated in the first place. When exploring the evolutionary history of most organisms, scientists can look at fossil records and similar historic evidence. However, viruses do not fossilize as far as we know, so researchers must extrapolate from investigations of how viruses evolve and by using biochemical and genetic information to create speculative virus histories. Most scholars agree that viruses don't have a single common ancestor, nor is there a single reasonable hypothesis about virus origins. There are currently evolutionary scenarios that may explain the origins of viruses, and if you have your own idea, feel free to enter the area of research. Your idea might be as good as anybody else's. This is a completely kind of uncharted area of biology. Now let's talk about the morphology of viruses. Morphology is a fancy word for shape, okay? Viruses are non-cellular. They are not cells, they are not living. These are biological entities that do not have a cellular structure. They therefore lack most of the components of cells, such as organelles, ribosomes, and the plasma membrane. A virion consists of a nucleic acid core, an outer protein coating or capsid, and sometimes an outer envelope made of protein and phospholipid membranes derived from the host cell. Viruses may also contain additional proteins such as enzymes within the capsid or attached to the viral genome. And these serve as machinery to get that genomic material or DNA or RNA into a cell to make it hijack the cell and create copies of itself. So there's lots of different parts and pieces to do that. And it's not very big. Viruses are surprisingly simple, even though we have this figure here that looks somewhat complicated. The most obvious difference between members of different viral families is the variation in their morphology, which is quite diverse. An interesting feature of viral complexity is that the complexity of the host does not necessarily correlate with the complexity of the virion. In fact, some of the most complex virion structures are found in the bacteriophages. Bacteriophages, remember, are viruses that infect the simplest living organisms, bacteria. Viruses come in many shapes and sizes, but these features are consistent for each viral family. As we have seen, all virions have a nucleic acid genome covered by a protective capsid. The proteins of the capsid are encoded in the viral genome and are called capsomeres. Capsomeres, okay? That's those are the proteins that make up the capsid. Some viral capsids are simply helices or polyhedral spheres, whereas others are quite complex in structure. This figure might be a little hard to see. Let's take a look at another one. And so here we see different sizes and shapes of things, okay? In general, the capsids of viruses are classified into 4 groups. Helical, okay? This tobacco mosaic virus is a helical one. Enveloped, okay? I'm sorry, icosahedral. The adenovirus is an icosahedral virus. You can see how it kind of looks like a kind of a jaggedy object. Enveloped, on the very left, this HIV virus is an envelope. And head to tail, okay? This bacteriophage here, the second one from the left, is a head and tail, all right? So helical capsids are long and cylindrical. Many plant viruses are helical, including the tobacco mosaic virus, which is a plant virus, right? Tobacco plants. Icosahedral viruses have shapes that are roughly spherical, such as those of poliovirus or herpes viruses. Enveloped viruses have membranes derived from the host cell that surrounds the capsids. Animal viruses, such as HIV, are frequently enveloped. We, again, are animals, right? Head and tail viruses infect bacteria and have a head that is similar to icosahedral viruses and a tail shaped like helical viruses. Many viruses use some sort of glycoprotein to attach to their host cells via molecules on the cell called viral receptors. For these viruses, attachment is required for later penetration of the cell membrane. Only after penetration takes place can the virus complete its replication inside the host cell. So you could think of these as parasites, okay? They need a host cell to complete their life cycle. I call it life cycle, but remember, viruses are not living. So maybe I shouldn't have said that. The receptors that viruses use are molecules that are normally found on cell surfaces and have their own physiological functions. It appears that viruses have simply evolved to make use of these molecules for their own replication. In other words, cells, like a human cell, for example, will have these little receptors that do their own thing. And then a virus will actually be able to come into that and trick the cell into, you are good, and take the virus particle into the cell. And then it will open up and release its genomic material and hijack the cell, and basically it becomes its home. I guess I should talk about the coronavirus. For those of you who are Hispanic, for example, you know corona means crown. Here's corona extra on the right, and you can see that little crown there. So coronavirus means crown because it kind of looks like a crown if you're looking straight down on a king. Those are kind of the spikes of the crown. And these are called spike glycoproteins. Those bind to receptors on the human cells, and then the human cell will recognize it as a good thing. Come on in. A welcoming party greets this virus, okay? And then all of these glycoproteins fall off, the membrane proteins fall off, and then this, it's kind of like a slinky. This slinky that's wound up here is the green on this figure, and that's called the nucleoprotein, okay? Which, I'm sorry, that's the genomic RNA it's pointing to here, okay? The genomic RNA. So that will be released, and then cellular machinery that's already in our cells will make copy after copy after copy of this, okay? Hundreds of thousands of copies, and then each one of those will come off the cell in a budding process, and then each one of those coronavirus particles will infect a new cell, and then each cell will make another 100,000 copies, and then you can see how it very rapidly divides. I wish I could talk more about this, but this is just a brief talk. Sorry about the slide here. This should be changed. But let's talk about 3 of these viruses here. One of the most complex varions known is the T4 bacteriophage there on the bottom left, which infects the E. coli bacterium. It has a head structure that the virus uses to attach to host cells. I'm sorry, a tail structure like a spider, okay, that's used to attach to host cells, and then the head structure up on top houses the DNA. There's an animation that you should watch for that. Adenovirus is a non-enveloped animal virus that causes respiratory illness in humans. It uses glycoprotein spikes protruding from its capsomeres to attach to host cells. Non-enveloped viruses also include those that cause polio. It's called the poliovirus, plantar warts, which is papillomavirus, and hepatitis A, which we call the hepatitis A virus. And that's top right, okay? That was the top right I was discussing there. Enveloped viruses, bottom right, such as the influenza virus that causes, the flu, consists of nucleic acid, which is RNA in the case of influenza, and capsid proteins surrounded by a phospholipid bilayer envelope that contains virus-encoded proteins. Glycoproteins embedded in the viral envelope are used to attach to host cells. Other envelope proteins are the matrix proteins that stabilize the envelope and often play a role in the assembly of progeny varions. Chickenpox, HIV, and mumps are other examples of diseases caused by viruses with envelopes. Because of the fragility of the envelope, non-enveloped viruses are more resistant to changes in temperature, pH, and some disinfectants than enveloped viruses. Okay? Overall, the shape of the varion and the presence or absence of an envelope kind of give us an idea about what disease the virus may cause or what species it might infect. But they are still useful means to begin viral classification. Okay. As I mentioned, you really want to check out this bacteriophage T4 animation. Here's the link. Click on the description in the video below, or if you're in my course, click on the Canvas link. That's it for the bacteria and Archaea. I hope you just have a bit of an idea about things here. Thank you for watching. Please like and please do consider subscribing to my channel. Have a great afternoon.